Hi, everybody. So, biology is very complicated. There's a lot of things that go into biology. And visualization of biological tissue has been a long-standing thing. Back in mid-17th century, about 1660 or so, there's an individual by the name of Robert Hooke. This was the first guy that looked through a microscope and saw cells for the first time. When he published his book, Micrographia, that was the first published circumstance of the term cell. And when he did so, he was looking at the cork cells behind us, these little clusters of cells that were definitely distinct things within this plant tissue. That was 400 years ago. Things have moved a little bit since then. This is the modern techniques that we use for visualization of biological tissue. That's a confocal microscope. It is able to do three-dimensional stacks, but only in thin volumes. In the image beside it is an axon. That's the growth cone of an axon. So we can get high resolution of these things at sub-micron levels very accurately, but it's still within 2 dimensions. One of the things that's missing is biology is actually happening in 3 dimensions. How can we visualize these things in 3 dimensions and be able to visualize them at whole volumes? We can look at small things in 3 dimensions, maybe hundreds of microns. For example, a single cell could be about 10 microns wide or so. So you're still looking at very small volumes of tissue. There's a lot of new techniques that have been looking at larger volumes of these tissue within 3 dimensions, but it's still not that large. Histology, in particular, is very interested in looking at tissue. Histology, the word comes from the study of the ology, and histo, which means tissue. In this field, they compare healthy tissue to unhealthy tissue or pathological tissue. By looking at those different types of tissue, you can identify this is actually healthy tissue, or in the example of oncology, you can look at this tissue using a stain like this and say, this tissue is diseased. It has a tumor in it, for example. This is cancerous tissue. If you go in and have a biopsy taken, this is what happens on the other side of it. They go in, apply this stain to it, can look at the individual cell nuclei, they can look at the tissue and say, yes, this is a tumor, or no, this is not a tumor. Again, they're still doing this in 2 dimensions. Biology is 3-dimensional. The process of doing this takes a long time as well. You take the piece of tissue, you embed it in some kind of material, you slice it and put it on a cover slip. And then once it's sliced and put on the cover slip, you put the stain on it. You can then put it under the microscope, then take a picture of it, and you're still looking at these things in 2 dimensions. This process can take weeks, months, years. People spend their entire academic career trying to refine this process, and they're still looking at it in just 2 dimensions. They're missing most of the tissue. So what the company that I work for is 3Scan. What we do is take look at tissue and do high throughput computational biology in 3 dimensions. We can take an entire organ, say a brain, stain that, put that in the embedding material, and then using our knife edge scanning microscope, we can slice that tissue, stack it, and then look at it in 3 dimensions. So let me take you through a quick example of how our technology works. We have the knife, we have the tissue that's on a stage, that stage moves along. We give a robot a knife, which is always a dangerous thing, but our robots have a diamond knife that they use to slice the tissue. This diamond knife is illuminated with a fiber bundle coming behind it. So as the knife slices the tissue, it illuminates the tissue from below. Then we have a microscope above that that is able to look at the tissue as we slice it and take pictures of it. These pictures are then recorded on hard drive. We can do this for large volumes of tissue, whole organ, for example, like an entire mouse brain, for example. One of the challenges that we've had, 3Scan is a complicated company because we've been innovating in a lot of different realms. Trying to get a stain into an entire organ is actually a difficult process. Like I said, the original techniques do it within a 2-dimensional slice. It's easy to get have the chemistry happen within 2 dimensions. Trying to get a molecule to dissolve into the middle of a brain, for example, is actually a complicated process. So we've been innovating in that way. Creating this robot, the knife-edge scanning microscope, has also been a complicated process, and we've taken years trying to get things to that point so we can collect this data. My job with 3Scan is looking at the data that comes off of the microscope and reconstructing it and being able to see what is in that tissue. This is an example of one of the strips of data that comes off of our microscope. It's really big. For example, you know your phone, the camera on your phone might take maybe a 12 megapixel image. That's that's a fairly big picture. There's a lot of resolution on that. This image ends up being about 12 megapixels, something like 3,000 by 4,000 pixels or something on that order. This image is about 4,000 by 70,000 pixels. It's about 75 megapixel, and this is just one small slice of it. We can zoom in and start to get a feel for what the tissue actually is within this slice. We can zoom in further, and we can actually start seeing the structures that are within this tissue. This particular stain is called India ink. It's looking at the microvasculature of an object. So one of the things that we're trying to do is look at the patterns of microvasculature across an entire organ. I'll have some slides coming up that will show that. So this slide describes most of what my job is. We take the large slices, clean them up, and stitch them together, as in figure B. You can see that there's some luminance artifacts, so we need to clean those up, and we can actually look at the tissue. You're starting to get the idea that this is actually a piece of the kidney. This is a 2-dimensional image of the kidney. Part of my job is to identify the portions of that tissue that are important, for example, the blood vessels. This is a segmentation or classification problem. We use a lot of different methods for this, including deep learning and a lot of other machine learning techniques to be able to identify these target objects. Given the identification of the target objects, we create 3-dimensional renders of these objects and then can perform a lot of different analysis based off of those 3-dimensional renders. Here's an example of another type of stain. So originally I showed you that H&E stain that was looking at the cell bodies and the tissue around it. This is one of a small portion of an image that we collected that represents about 5 millimeter by 5 millimeter piece of tissue. Most histology is looking at slices that are about 1 millimeter by 1 millimeter. This is actually a small image for us. From this, we can actually create a large-scale render of this tissue. So with these techniques, we can actually start to look at the 3-dimensional structure of tissue at unprecedented detail, at micron level resolution. This allows us to go in and see, yes, this is actually a tumor with great fidelity. This can be used in drug screening. This could be used in a lot of different techniques for 3-dimensional analysis. This is an example render of that kidney that I was showing you. From the 2-dimensional images that we capture, I'm able to collate them and create this 3-dimensional render of this. This image has been downsampled by about 10 times. We have full resolution of this image, gets us very tight representation of the microvasculature. Here's another interesting one. This is half of a brain. This is all of the microvasculature within a mouse brain at a high resolution. So as you can see, just even within this brain, there's a lot of different types of microvessels that occur within this brain. One of the things that we're working on is trying to create metrics off of these microvessels. If we're looking at the frontal lobe, what's the pattern of the microvessels within that area? If we're looking at the cerebellum, what's the pattern of microvessels within that area? If there's a tumor there, tumors are known to have different microvasculature, so we can quantify those differences of what normal microvasculature is and compare that to the tumor tissue and say, yes, this is in fact a tumor. So that's a full zoom out. That image was downsampled a lot to create that render. This is from the same images, but a lot closer. So you can start to see the different scale of structures that occur within all the data that we're able to capture. This is from the forebrain. This is from the frontal lobe. And as we fly in, you can see that there's the major vessels that come through. Each of those is maybe 100 microns in diameter. And there's this cloud of all the microvessels that are 10 microns in diameter. We're able to capture that network as well, this plexus of vasculature. From that network, we create a lot of different other algorithms that are able to trace the center lines through those networks. And from that, we can calculate the length of each of the vessels, the number of branch points, the tortuosity, how much the vessels wiggle around within the brain. Tumors are known to have a high tortuosity in their vessels, for example, and then start to compare the different structures. This last image is of the pancreas, and in comparison to the brain, you can see that it has a very different type of network structure. It's more tree-like. It has a lot of branches, as opposed to the brain, which has a lot of loops. And again, we're able to quantify these differences using graph theory to be able to trace the differences of these networks. So using the techniques that we have, we're able to add this new dimension of tissue imaging and be able to look at an entire organ and quantify that organ in unprecedented detail. So what's next? A couple of the future things that we want to do is look at the output from this slice. We can capture the slice after we image it, and once we capture it, we can do a microdissection. We know that there's this fluorescent label on it, and we can dissect out whatever the tissue is that has that label and pass that to a gene sequencing machine, for example, and be able to identify the genes that are labeled in that area. There's a lot of other imaging modalities that exist that we can use in this technique as well, such as fluorescence. In general, the techniques and innovations that we have expanded out the data from a single image that could be 12 megabytes to that volume of the brain, which is 1 terabyte, and we collect one of those a day. That's a lot of data that has a lot of impact and has the ability to change medicine as we know it and be able to process these in rapid manners. And I think that's it. Are there any questions?
I'm just an interested layman, but who writes your code? You write the code? Oh, well, you're my new hero. That's the end of my question. Thank you.
All right, other questions?
You mentioned that this helps identify tissues and tumors, but would it help you identify if something has a malignant tumor or the malignancy of a tumor, or does it not help with that process? And also, how does this help with identification of tumors to disease prevention or disease in that element?
Thank you for that question. So in two-dimensional histology, they can look at the different structures within that, the cell count, the irregularity of the cell body. And through those things, they can identify whether it could be a benign tumor or a malignant tumor. And through the structural components, through these classical techniques, they can identify those things. And again, one of the benefits that we have is rapid throughput. So if we have these different pieces of tissue that we get, then we can put it through our microscope and be able to identify the features of what a malignant tumor is very rapidly, and then pass it to a histologist for confirmation that our prediction is correct. One of the collaborations that we're working on, many of you might know of the Mayo Clinic in Rochester, Minnesota. We are working on a collaboration with them. They are the world's largest recipient of tissue from everywhere to be able to do a classification. They have a massive pipeline of histologists to be able to receive that tissue, stain it, slice it, image it, and identify whether it's a tumor or not. What we can do is take that process and condense that down to a day to be able to do that classification and then pass it to a histologist for confirmation. So yes, it speeds up the process a lot.
I apologize, I came in a little late on your presentation. Back to the configuration of blood vessels in the brain and the recognition that the configuration of a tumor is different. Can you differentiate, say, artery from vein? The passage. What I'm leading up to is, having recognized this configuration, especially with the tumor, is it possible to selectively block the blood vessel entry and starve out a tumor?
It might be. Identifying an artery versus a vein, there's a lot of different techniques that you can do that. Arteries tend to have smooth muscle around them, while veins are just big sacs. So you can look at the muscle content around the vessel and see whether it's an artery or vein. It's still a loose feature that is associated with that. That identification of artery versus vein is a very important metric for a lot of different things. For example, tumors have, one of the metrics that I'm using is vessel diameter. There's a distribution of vessel diameters that are expected within the brain that follows this power law. Tumors violate that power law very quickly. And if you have a large artery that's coming in and suddenly branches out into a million different microvessels, that's definitely a very different pattern. Being able to identify the vein coming out of that tumor, again, the perfusion of a tumor ends up being a big concept because they require a lot of blood to be able to grow the way that they do. Blocking off the artery, I mean, our technique is destructive, so I mean, it kind of doesn't matter. we perfuse the animal and then fix the tissue before we slice it. So it's definitely different than, say, an MRI, which is in vivo. But one of the, actually, we have a couple collaborations that are going on right now that are doing in vivo studies using MRI or PET scans, and which have voxel resolution of about 1 millimeter, and then we have our voxel resolution of a micron. And so we can take our data and overlay it with whatever their data it looks like from the in vivo, and that gives new models of prediction of blood vessel flow from all of these large non-invasive imaging techniques, and improves the models, predictive models of these different things, and potentially ways to address blood flow within that tumor.
I hope I didn't miss this part of the talk. How well can you model the rest of the organ or tissue that you're looking at when you're not able to collect that data from the sample? Like if the sample is damaged, can your code predict the rest of it without actually having that information?
I'm not sure I understand. So not having the information from the image that we collect, for example, like if we have an image like this,
right,
there's a lot of information in there. I'm creating features to be able to identify things. And I think your question is asking if my features aren't capturing all of the data.
Correct. So if you have a tissue, you basically layer it to make your three-dimensional object. But at the end of your images, it's basically a flat surface because there was nothing else for you to capture.
Right.
Are you able to predict from the information that you have accumulated how the rest of the tissue could possibly look? So if you're looking at tumors,
of disease tissue.
Disease tissue.
I mean, yes, there's definitely some consistency of the statistical models of these things. Like if you take a look at a liver, like there's distinct patterns within the liver and how those cells. I don't think I have any liver images with me here, but you can make inferences from those different things from the statistical properties of it. It's one of the differences of stereology versus tomography. Stereology looks at a couple slices of a thing and says, yes, there's this cell count in this particular area, and in this other area there's this many cells, and we can assume that everything in between has about that kind of cell count. Tomography looks at all of the data and makes no assumptions about the statistical properties of it. It just actually does the count for those missing areas. What we do is more along the lines of tomography, so we try to minimize the missing information from our system.
All right. What an awesome talk when you just jumped right in. Thank you.